Hello, everyone. If I don't use the cube, if I just yell like this, can you guys hear me? Is this okay? Sweet, because the cube is awkward. All right, so thank you everyone for coming to seminar today. I am so excited to have our friend and collaborator and absolutely awesome scientist, Dr. Blair Perry here. Blair is one of probably the coolest scientists I know because he works on such a wide variety of really awesome systems. Basically, his work focuses on the genetic basis of complex adaptations, and he's worked on everything from rattlesnake venom to bear hibernation to extremophile fishes, and even has dabbled in blood flukes and COVID publications. He's kind of done a little bit of everything. So even though he got his bachelor's from The Ohio State, that's fine, we'll forgive him. Don't do that. Which is actually where he and Matt met. He did his PhD at UC Arlington with Todd Casto and worked on kind of two different projects there, worked on genomics and rattlesnake venom evolution, and then also studied gene regulation and expression of molecular pathways that are responsible for post-feeding organ growth in pythons. So if you don't know a lot about snakes and they eat a big meal, their heart and stomach physically grows more tissue. He studied all the cool gene regulation things that make that happen. And this is actually where I met Blair when I did my first field work here at Michigan. I went to Texas to collect. Blair came out and helped me round up water snakes and cottonmouths and all the things in the drainage ditches around Dallas. So that was our first field work foray together. After getting his PhD, he went on to do two postdocs, one at UC Santa Cruz and one at Washington State University. Both of these focus on the signaling pathways that underlie bear hibernation. And as of last year, he is now our new neighbor at Michigan State University. So if you like what he's talking about, chat with him more. He's going to be around for a while. And with that said, please join me in welcoming Blair Perry.
All right, thank you so much. Can everyone hear me okay? Volume, feedback okay? I feel like I'm feeding back a bit. All right, awesome. Well, thank you, Haley, for that nice introduction. Thank you all for being here. Thank you to everyone who helped organize this visit and for the folks that I met with and chatted with today. It's been really awesome to see all of the really cool and diverse research being done in this department, to see this incredible building and some of the museum. It's been a really wonderful visit so far. So as Haley mentioned, I do a lot of work in a variety of things. My lack of being able to pay attention on one specific thing, I guess, can also be seen as a fun thing or a cool thing about science. But today I'm going to try to focus on one thing, which is my recent work and some directions that I plan to continue on studying the gene regulation and physiological adaptation in bears with a focus on hibernation. So, a fundamental goal in biology, and one of the very basic goals that drives a lot of my research interests, is trying to understand how genotypes, genetic information in an organism, manifest as phenotypes. And one of the things that I think makes this really interesting, but also a bit of a challenge, is that between genotype and phenotype is this box containing what we refer to as gene regulation. So gene regulation is a complex suite of genomic sequences, genes, signaling molecules, interactions between all these different things, and more that ultimately control when, where, why genes are expressed in different cell types, tissues, etc. And because gene regulation is kind of translating this genotype into phenotype, if we modify aspects of various components of gene regulation, we can sometimes modify a phenotype without necessarily changing the underlying genotype. And this can happen at multiple different scales. So this can happen over evolutionary time as these signaling pathways and interactions evolve and change. This can happen across and within populations, leading to phenotypic diversity, like shown in these butterflies here. And at a very fundamental level, this is happening in every cell in your body. This is the way that we have one genomic sequence that produces all of these specialized cell types, tissues, organs, etc. So the real big kind of general question that drives a lot of my work is understanding how things that fall into this box of gene regulation, how the evolution of these gene regulatory mechanisms drive adaptation and result in variation in nature. So as Haley mentioned, I just recently started my lab at Michigan State. I started in January, so I've been there just over 3 months. So this is currently everyone in the lab. It's just me, but I hope to have a few more people in this photo by the end of the year. My lab is aimed on understanding the evolution of complex adaptations, particularly the role that gene regulatory architecture plays in the evolution and modification of these adaptations, and trying to integrate functional genomics, evolutionary genomics, and population genomics data to understand adaptations in kind of multiple different dimensions or from different approaches. I also study snakes. I'm not going to talk about any of that work today, but I felt like I had to have a snake in here somewhere. So for my talk today, a brief overview. I'm going to give you kind of a quick refresher on gene regulatory architecture, just for those of you who might not have thought about this in a while. Introduce you to all this cool biology that falls underneath mammalian hibernation, and then talk about some recent work looking at the regulation and evolution of hibernation in bears, looking at regulatory sequence evolution and its role in hibernation, and then teasing some kind of more recent and continuing work looking at population genomics and trying to understand hibernation using population genomics of brown bears. And then I'll try to wrap it up in some sort of conclusive or cohesive conclusion at the end. So starting with the introduction, bear with me as we go through some gene regulatory concepts and terms before we get into the cool hibernation biology. I just want to kind of make sure everyone's on the same page and get some textbook definitions out of the way here. So one of the major or common components of gene regulation or gene regulatory architecture are cis-regulatory elements. These are sequences or regions in the genome that are bound by transcription factors and other molecules to regulate one or more target genes. The two most common or widely studied classes of cis-regulatory elements include promoters. These are the site of transcription initiation. They are typically located just upstream of the gene that they regulate, and they are bound by RNA polymerases and other machinery to initiate the transcription of their target gene. Enhancers, on the other hand, interact with promoters to control the expression of one gene, potentially multiple genes in some cases. But unlike promoters, can be located really, really far away in the genome from the gene that they target. This can be thousands of base pairs, hundreds of thousands of base pairs or more in some cases. And they come into contact and interact with the promoter and associated transcription factors through these three-dimensional changes in orientation and interaction of the genome. Transcription factors, a term I've mentioned a few times, are referred to as transregulatory elements. These are essentially proteins and other molecules that are also involved in this process, for example, by binding to specific small sequence motifs within enhancers and promoters and other cis-regulatory elements to help contribute to when, where, why, and how much these genes are expressed. And then kind of more of a conceptual definition or concept here is these signaling pathways and networks. So these are cascades of interactions between genes, proteins, other molecules that can typically translate some stimulus or high-level molecule into a gene expression response, other cellular processes, other kind of responses to a stimulus. So in a very general sense, these are kind of the wiring that connects things in the cell and makes biology happen. So those are my gene regulation definitions, trying to keep it brief and not too boring. There's a lot more that goes into gene regulation than just this, but I won't get into that today. But just know that there's a lot of complexity, which makes this really fun and really exciting to understand, and also challenging in that understanding all of these different applications or aspects of gene regulation are often necessary to understand how genotypes are translated to phenotypes. All right. Let's get into hibernation. So there's a lot of cool biology related to hibernation here. I'm guessing that most people are at least somewhat familiar with the concept of hibernation. This is an adaptation broadly to conserve energy during periods of resource scarcity. Oftentimes this can be associated with the winter months, but in some systems is in the summertime or other times where for some reason or another there's a reduced availability of resources for a prolonged period of time. And typically in mammals, this involves a complex suite of physiological changes and shifts, such as a reduction in metabolic rate, a reduction in body temperature, limited feeding, complete fasting in some species, and generally prolonged periods of inactivity or something similar to sleep. And this is a tree of mammals over here, or some representation of mammals. And as you can see in blue, there are forms of hibernation that kind of pop up all throughout this tree. And if you look at the species that are associated here in blue, typically these are small mammals like rodents, some bats, a few small lemurs hibernate. And then down here at the bottom you have bears, which are by no means a small mammal. So it turns out bears are actually pretty unique when it comes to hibernation within mammals. So they exhibit a lot of unique features that make them a very kind of distinct form or flavor of hibernation. So typically when we look at hibernation in small mammals like ground squirrels, for example, and we track something like body temperature over the duration of hibernation, we see these really drastic drops in body temperature, sometimes reaching essentially environmental levels, so near freezing. But you see these periodic spikes where everything essentially comes back to almost a normal body temperature, metabolic rate, and so on, bringing everything kind of back online, back up to its full form and function, so that everything can stay functional, healthy, and so on before this next drop into this deep period of torpor here. So there are these inter-torpor periods of arousal here. In contrast, bears do kind of a low and slow approach. So shown here in blue is the body temperature, representing a bear over the course of hibernation. And the drop is not nearly as extreme, but they maintain this reduction in body temperature, metabolic rate, and so on for the duration of hibernation, which can last up to 6 months in some cases. So, not as deep of a drop, but they're sustaining this the entire time. They have this, again, kind of prolonged torpor period with no periodic arousals. There are some other cool features that were initially thought to be kind of defining features of hibernation, such as the loss of circadian rhythmicity and gene expression and other cellular processes in small mammals. Bears maintain circadian rhythms throughout hibernation despite being in a den without access to daylight cycles and so on. And they also just have a substantially larger body size than we typically see for mammals, which suggests potentially some unique kind of metabolic constraints and demands for these large animals compared to something like a small ground squirrel. So thinking a little bit more about hibernation in kind of the annual sense, we can split it up into these 3 different seasons or periods of the annual cycle that are all equally important to hibernation itself. So the first season being the active season. This is in the spring as bears emerge from hibernation, and they're doing just kind of normal bear stuff. So they're dispersing, they're eating, berries, things like that, gaining kind of lean mass. They're mating during this period of time. And this happens kind of from late spring into late summer when they enter this hyperphagia season. Hyperphagia just means essentially like obsessive eating or hyper eating. And during this time, bears truly become behaviorally, physiologically set on eating as much as possible, gaining as much weight as possible in a short period of time in preparation from hibernation. So some of you may have seen like the Fat Bear Week outreach stuff that happens every year. This is happening during hyperphagia as bears are gaining as much weight as they can. They're consuming, in some cases, over 20,000 calories per day, gaining over 10 pounds per day in some cases. And hopefully those numbers are pretty impressive. But for a visual aid, this is a bear at Katmai National Park, nicknamed Holly. This is Holly in July of 2019. So she's already gained some mass back after exiting hibernation. This is the exact same bear in September of the same month. It's hard to believe that's the same animal and just like 3 months later, but that is indeed the same bear. Holly is clearly doing a great job getting fat in preparation from hibernation here. And the reason that this is so important is because gaining all of this fat is how they are able to sustain their metabolism and homeostasis throughout hibernation. So they shift to primarily lipid metabolism during hibernation, burning this fat, sustaining metabolic and physiological processes. And therefore, the fatter they get in the fall, the more likely they are to have a healthy hibernation season. A fun fact I'd like to throw in here, because I think this is a common misconception of hibernation in bears in particular, is that they're not necessarily sleeping the entirety of the winter. In their dens, they will move around, they'll readjust, get up, kind of walk around, sometimes drink melted snow at the entrance, things like that. But they're by no means like in like a coma-like state, which you do see in some of the small rodent hibernators. So that's a fun trivia fact to take home. But there's a lot of really cool stuff that's happening during hibernation. It's still a very extreme thing that they're experiencing every year. So there's a 75% reduction in metabolic rate, a 4 to 7 degrees Celsius reduction in body temperature. Again, not as extreme as those rodents, but still very extreme for a large mammal. Heart rate can slow to as low as 10 beats per minute for, again, like 6 months out of the year. And they don't feed, they don't urinate, they don't defecate, and they drink essentially no water, but there have been a few anecdotal instances of them drinking. And this is also my reminder to have a sip and take a breather. All right, so if you look at this list of kind of cool and extreme features of hibernation and think about if a human were to experience any one of these things, let alone all of them, it would essentially be like a very severe medical emergency, right? There's all sorts of really cool aspects of hibernation that have these really direct kind of parallels or rather kind of defiances of what we think of as kind of typical for human biology. I'm not going to focus on it too much today, but just to highlight one of these features that we're interested in from kind of the translational side as well. Bears become insulin resistant during hibernation. Specific tissues within the bear actually become insulin resistant while others retain insulin sensitivity. Insulin being this hormone that helps signal and regulate glucose uptake, the cells essentially no longer respond to it. But they regain the sensitivity every single spring kind of automatically as they exit the den and kind of re-emerge into the active season. In humans, insulin resistance is typically a very, very big deal. This is an early indicator of the development of type 2 diabetes, and typically it's really difficult to kind of regain insulin sensitivity without medication, really severe changes to diet, exercise, things like that. Bears are essentially emerging from the den, and it's just kind of all returning to them without any additional effort. And again, I won't focus on the translational side too much, but essentially every single tissue that's been studied in bears thus far has some really apparent and cool translational potential for understanding, how human diseases happen in the first place, why bears are able to experience all these extreme changes year after year without harmful consequences. So a lot of work being done in the hibernation realm is trying to understand, how does this work in bears? How can we apply that to treat or prevent disease in humans? Well, let's get back to the bear biology. So understanding this complex physiological adaptation at a gene expression level has been a relatively recent endeavor. The first big paper in this came out in 2019 and showed that this pretty extreme physiological shift is driven by a pretty extreme gene expression response as well. So this study looked at 3 different tissues, metabolically relevant tissues, adipose, fat tissue, liver, and muscle, and found in total across these 3 tissues, I think a little over 10,000 unique genes in the bear genome that are significantly differentially regulated or expressed between the active season and hibernation season. So like roughly half of the genes in the bear genome in just these 3 tissues. And when we look at what these genes are involved in, you kind of see exactly what you would expect in terms of the regulation of metabolism in a mammal. So genes involved in insulin signaling, lipid metabolism, processes or pathways involved in muscle degradation and preservation of muscle tissue, and more. Which I think is really cool because, unlike some adaptations where you have a specific set of genes that have evolved specifically to facilitate or drive like some unique phenotype or physical structure, bears are essentially using, or it appears to be using, the same genes, the same pathways, essentially the same toolkit present in humans and almost every other mammal and vertebrates more broadly, but in this very unique way to achieve these feats of hibernation. So kind of using a common set of tools in a unique and special way. The last thing that I want to kind of highlight within this crash course on hibernation biology in general is also kind of another fun fact, which is that not all bear species hibernate. So brown bears in particular have become kind of this poster child in pop culture for hibernation and things like that, but it turns out a lot of bear species that inhabit environments, closer to the equator that don't have such strong seasonality do not need to hibernate because they have resources year-round. Where in fact the American black bear, brown bear, and Asiatic black bear are the only species that have this really drastic annual hibernation cycle. Polar bears are a very interesting case here, shown in yellow, and we kind of refer to them more recently as facultative hibernators, and that they essentially can hibernate in certain conditions when they need to. A little bit more information on this will be relevant later. They don't have this really drastic annual cycle like we see in the brown bears. Some large males have been shown to hibernate actually, or something similar to hibernation in the summer when food is scarce for these bears. Females will also den up when they're pregnant, and we think they do something similar to hibernation, although it's still not fully well understood. But there's also, more broadly, a lot of really interesting metabolic and physiological adaptation that's occurred in polar bear. They're fully carnivorous. They have a higher resting metabolic rate than other bear species, and inhabit this unique Arctic environment with a lot of unique energetic and metabolic demands. And what I think makes this even more exciting from a biological perspective is that they are extremely recently diverged with the brown bear, about only 500,000 million years estimated divergence from the brown bear, which is again our poster child of kind of this extreme hibernation adaptation. So a lot has happened in a very short period of time in the evolution of the polar bear. I'll circle back to this in a little bit. But broadly, my research is interested in understanding hibernation kind of from as many axes as we can understand. So what are the underlying genomic mechanisms? What is the gene regulatory landscape that's enabling these bears to use existing genes and pathways in this unique way? What signaling or high-level signals control these signaling or shifts in expression in physiology as bears are entering, exiting hibernation, and more? And broadly, how did hibernation evolve across bears, mammals, and vertebrates more broadly? Last thing that I want to touch on before I get into some of my recent work is that a lot of this work has been done through collaboration with the Washington State University Bear Center. This is a very, very unique facility in Pullman, Washington that has a captive population of 11 brown bears that we are able to study safely and in a controlled way year-round, so we can anesthetize bears with the full vet staff support, take samples during hibernation, take metabolic and physiological data year-round, all this really cool stuff that would be literally impossible or at least extremely difficult in wild bears. And there's a whole lot of cool bear biology going on here related to the physiology and genomics of hibernation, population genomics, conservation, diet, nutrition, energetics, and so on. And just kind of as a fun fact here, this bear here you can see is presenting a leg through this opening here. They're trained to do this so that we can give them medicine or take a blood sample in exchange for a squirt bottle full of, you might guess this, honey and water. They're very treat motivated, so they are very quick to pick up on these behaviors that allow us to study them minimally invasively, and they get a nice honey treat as a response. All right, so let's get more into the recent work here. So I'm going to talk a little bit about kind of two areas of research that I've been working on recently, looking at the regulatory sequence evolution underlying hibernation in bears, and then a little bit on the population genomics of North American brown bears and how we can use this to try to further understand hibernation. All right, so I talked about this a little bit earlier, but the reason that I think regulatory sequence evolution is really interesting in the case of hibernation is that by modifying aspects of the gene regulatory architecture, you can, in theory, alter expression or kind of rewire or retool these existing regulatory networks and interactions in a way that you can tweak how a gene is behaving, when it's expressed, where it's expressed, how much it's expressed in a particular context. This could be a tissue, a time point, a life stage, a season in the case of the bears, but without disrupting its activity and other contexts where its previous function is needed to be kept. And this contrasts to like protein coding changes where you're changing the sequence of that gene across every tissue, every cell type, potentially disrupting its function in cases where it needs to stay how it was. So, there have been some that have posited that perhaps regulatory evolution for this reason has less strong of constraint compared to protein coding sequence changes. This is still kind of up for debate, but regardless of that specifically, there's been a lot of work that's shown in mammals and other species that enhancers in particular appear to evolve rapidly, and that they appear to underlie a lot of really interesting, complex, expression-driven traits across a diversity of species that have been looked at. And in the case of hibernation, this may be particularly relevant because we have thousands of genes, again, that are being differentially expressed every year across these multiple tissues in a coordinated manner. We appear to have non-canonical or kind of atypical regulation or activity of some of these conserved and prominent metabolic pathways that we see across mammals and vertebrates more broadly, like insulin signaling. And we also have these cool instances where hibernation appears to have been rapidly lost or at least substantially modified in a short period of time, where perhaps modifying gene regulatory elements rather than the protein coding sequence may enable this to happen quickly as organisms are adapting to unique metabolic needs, for example, in the case of the polar bear. To understand regulatory sequence evolution and its relevance in hibernation, we need to know what the regulatory sequences are. And when this work started, we knew where the genes were. We had a nice genome. We can find the promoters. They're just right next to their genes. But the enhancers are a lot harder to find. Again, they're really far away from the genes in some cases that they're controlling. And in addition to this, they can have really tissue-specific activity. So if you don't have the right data in the right tissue, you might not be able to detect there's an enhancer, being active in a particular place in the genome. And they appear to evolve rapidly. So, potentially just comparing sequence between known enhancers and another species in your species may or may not be effective. But they're also really difficult to generate, well, so typically you generate kind of layers of these functional genomic approaches like ATAC-seq, which measures chromatin accessibility, ChIP-seq for histone marks and things that are associated with the kind of the regions around these enhancers, and layer that up to identify putative enhancer regions. None of these approaches work well for adipose tissue, which is where a lot of our work is focused. It's one of the tissues we can easily access from these bears because they have a lot of fat. But that fat makes these protocols really difficult to optimize, takes a lot of time, a lot of samples, and a lot of money to do this. And despite having these large fat bears, we can only take small biopsies at a time, so we haven't really been able to perfectly optimize these yet. So we were trying to get creative with an approach to identify enhancers in the bears to do this work. And we landed on this cool, relatively new technique called capped small RNA sequencing. This is going to get a little kind of methods heavy for like a few slides, so just bear with me. Just trying to give you a little bit of context for how this approach works and how we used it. Happy to talk about the details of this more after. But just to give you a basic sense of what this does, this approach was designed initially to identify sites of transcription initiation in the genome, so essentially where transcription is happening in the genome at very precise base pair level resolution. And it uses total RNA as input. So if you can extract RNA from a tissue, which we can do for our adipose tissue, we can, in theory, use this approach. How does this help with enhancers? Well, so this approach captures a diversity of these short, nascent, or kind of young transcripts that are actively being transcribed. And in that mix of transcripts that it captures are enhancer RNAs. So enhancer RNAs are short, unstable transcripts that for the most part are thought to be kind of a byproduct of transcriptional machinery happening to bind to an accessible enhancer, transcribing a little bit of it before moving on to look for a protein or something like that. So typically these tiny little enhancer RNAs just kind of get kicked off from these regions, degrade, and don't do anything. There are some instances where they appear to play regulatory roles, but for the most part it appears that a lot of them are just kind of a random byproduct here. But while most approaches like mRNA-seq don't capture these transcripts, this approach does, and if we find them within our reads, we can map them back to the genome and use them to identify enhancer regions, just like you might map mRNA transcripts to find protein coding regions in a genome. The other cool thing about this approach is that it is, an RNA sequencing approach, which means there's a quantitative element to it as well. So we can actually get quantitative information about enhancer activity based on the abundance of enhancer RNAs that we're picking up from different regions in the genome. So we can use the counts or expression of these enhancers to assess relative activity levels of enhancers between different seasons or samples in our case. And also, given that these enhancers correlate with, or these enhancer RNAs correlate with enhancer activity, typically they also correlate with the expression of the target gene. So if an enhancer is more active, the gene that it is involved in activating is also going to have higher expression in the mRNA-seq data. So we can try to use this to correlate genes with enhancers that are relatively nearby and have kind of similar enhancer RNA and mRNA-seq expression. So in short, we can use this to identify enhancer regions, quantify relative enhancer activity, and identify potential target genes of these enhancers, all using RNA as the input and not having to worry about ATAC-seq and ChIP-seq and all these other trickier approaches for our bear samples. So we generated this data for a relatively small, but for bears, pretty large set of samples. 6 individuals sampled in the active season and in the hibernation season as well. This is all adipose biopsy tissue, so fat tissue where a lot of this cool metabolic shifting is happening. Extracted RNA with the typical RNeasy kit and generated both csRNA-seq data to measure enhancer activity and mRNA-seq data to measure gene expression. A few more methodsy technical slides here, and then we'll get back to some more cool hibernation stuff. By kind of filtering the transcripts and the pileups of read mapping that we get from this data based on expectations of what we expect enhancer RNAs to be doing, we expect them to be far from genes in the genome or kind of in intergenic regions, and we expect them to be unstable. There are ways that we can assess whether transcripts are stable or unstable using this approach, which I can talk more about later. We were able to narrow in on a set of loci about a little over, or I guess a lot over, 26,000 putative enhancer regions throughout the genome. And since this is quantitative, again, we can also compare the enhancer RNA expression in these regions between our active and hibernation seasons to narrow in on just over 1,000 enhancer regions that have evidence for shifts in activity level between the active and hibernation season. So this is kind of our full set of putative enhancer regions, and these we have additional layer of evidence that they are potentially actively involved in regulating gene expression during hibernation due to these changes in activity. And based on correlating, again, the enhancer RNA expression from csRNA-seq data with relatively nearby gene expression data from the mRNA-seq data, we were able to pair up about 3,000 putative enhancer to gene regulatory relationships. So essentially trying to find genes that we think these enhancers might be targeting in the bear genome. All right, all that to say, we didn't know where the enhancers were in the bear genome. Now we have a decent sense where some of them are, at least ones that are involved in adipose tissue gene regulation. So there's a whole lot of other functional kind of nitty-gritty stuff that we're doing to try to further vet these enhancers and validate them and so on. But with these enhancers in hand, we can now start to better address some of these questions about regulatory sequence evolution, how these enhancers might be changing across bear evolution, and how that might relate to hibernation as a cool and interesting adaptation. So I'm going to talk about kind of two very general hypotheses here. The first one being that hibernation evolved in part through the co-option or repurposing of existing gene regulatory architecture. Again, this idea that they're using potentially existing enhancers, existing signaling networks between these enhancers, genes, transcription factors, et cetera, in a unique way to achieve these feats of hibernation. And then also this hypothesis that the evolution of these enhancer regions may be particularly important for some of these instances of rapid loss or modification of hibernation and associated traits within bears specifically, for example, the polar bear with its recent divergence from the brown bear over here. So we're going to start with this one up here, and I want to just preface this by this being kind of a very coarse and exploratory, but I think kind of exciting, first look at this question. So I don't have a perfectly definitive answer here, but hopefully we'll get you excited about where this work can go. So again, the idea here being that hibernation appears to leverage these broad, conserved mammalian metabolic pathways in a unique way. Perhaps enhancers have been co-opted to facilitate regulation in bears in a unique way than what they typically do in other species. So essentially, we want to compare these enhancers we've identified in bears and what we think they're doing in terms of functions, what genes we think they're regulating, with kind of what homologous enhancers are known to do kind of in a normal species, you might say, like humans. So this is a very coarse comparison again, but humans have, maybe mice as well, but humans have probably the best characterized enhancer landscape. So identifying where they're at in the genome, identifying what tissues they're involved in, and identifying the genes that they target. Using this helper approach, we identified orthologous regions in the bear genome and the human genome, specifically for these putative bear enhancers. So finding orthologous regions in the human genome, and then compared those regions with this gene enhancer database, which is a large combination of multiple databases containing known human enhancers that have been discovered and validated through a variety of approaches, and also includes what we think or know are their target genes in humans in various tissues. Again, basic idea being are there enhancers that are conserved between these species? Are they targeting the same genes? If not, maybe that's interesting or relevant for hibernation. So walking through the results of this for this set of the full enhancer set to begin with, so over 26,000 enhancers, we see that about 70% of these bear enhancer regions have an orthologous region in the human genome. Of those orthologous regions, the vast majority of them overlap with known human enhancer regions in this database. This is cool, also a nice validation that this csRNA-seq approach, which we're kind of using in a way that it wasn't perfectly designed for, does appear to be pretty good at finding things that at least in other species are real enhancer regions. But when we look at these enhancers that are orthologous and also known to be enhancers in humans, we see that over half of these are inferred to be targeting the same genes in humans as we were inferring in bears. So, kind of conserved function in addition to conserved sequence. But there's a relatively small set here, about 11%, that are conserved in sequence or, orthologous between bear and human, but potentially divergent in function, in that the genes we're inferring them to target in bears are not known targets of these enhancers in humans. So this is, again, all the enhancers. There's probably some false positives in here. When we focus on that set of enhancers that we did the differential activity analysis, so we have more evidence for them being relevant for hibernation, we see pretty similar percentages or proportions of regions with orthology to humans and regions that have, are orthologous to a known human enhancer. But when we look at the genes that these regions are inferred to target, we see a substantially higher proportion of orthologous enhancers in bears that are, at least in our case, predicted to target genes that are not known targets of these enhancers in bears. So again, conserved in sequence and orthologous, but potentially divergent in function with a higher proportion of these kind of divergent, functionally divergent enhancers in the subset that we know are likely relevant for driving hibernation, aspects of hibernation gene regulation. And this is all kind of, cursory, preliminary stuff, but when we look at the genes that are inferred to be kind of these unique targets of these enhancers in bears, we do find genes that are really kind of central and important to some of these pathways that we know are relevant for driving metabolism in hibernation, like insulin signaling and PI3K, which are related pathways, but unique molecules within these two pathways are included in the set. We're still kind of parsing through these and trying to make some conclusions from that. But again, very kind of broad level analysis, but I think kind of a cool first look at this, suggesting potentially that there is evidence for rewiring of these conserved enhancers to target genes that are unique in bears, potentially relevant to hibernation. So non-canonical target genes of these conserved enhancers, some of them involved in these key pathways. There's a ton of caveats here. The bear enhancer gene relationships that we're inferring here, there's probably a lot that we just got wrong, maybe potentially inflating this number. So we're working on validating these further with additional layers of data. There's potentially some enhancer gene relationships that maybe we identified in bears that haven't been studied or identified in humans. And obviously this is also an extremely coarse level of comparison. We can't really say whether these functionally unique enhancers are specific to hibernation or just divergence in enhancer function that we're observing between bears and humans, which are like 96 million years diverged or something like that. So I'll take this with a grain of salt, but I think it's kind of a cool first step in this direction, and we're working to incorporate like mouse data and try to fill in some of the gaps here. I want to shift to focusing on bears a little bit more, and this idea that potentially regulatory sequence evolution may be particularly important for these instances of rapid metabolic and physiological adaptation within this group. So hibernation has rapidly been lost or modified throughout bear evolution. It's still kind of up for debate, but the consensus for the most part is that some form of hibernation was present in the ancestor of bears, and likely kind of specialized and lost and strengthened in certain lineages as bears evolved. But in these situations where we have apparent loss, has selection on enhancers and potentially promoters driven this physiological adaptation, enabling rapid metabolic and physiological adaptation in a very short period of time, for example, in the instance of the brown bear, polar bear divergence here. So the methods for this analysis were kind of similar to start. We identified orthologous regions across all of the 8 extant bear species using a cool cactus alignment that was published last year. And then we used a tool called PhyloP to assess branch-specific acceleration and constraint. So if you're looking at evolution in protein coding regions where you have a code, right? You can look for non-synonymous, synonymous substitutions and so on. You can't do that in non-coding regions because there's not a code to define those different types of substitutions with. So a lot of these regulatory evolution analyses rely on estimates of rate in these regions. And the idea here being that if we see acceleration rates or regions evolving really rapidly, these may be indicative of positive selection acting on these regulatory regions. And the opposite side of things, if we see constraint or really slow evolution, this may be indicative of purifying selection. We did this at a branch-specific level, or at least that I'll talk about today. So we're also looking for, on a particular branch, is there acceleration that's relatively high in that branch compared to other branches in the tree. And I'm going to focus on acceleration today because we didn't find a whole lot in terms of the constraint and purifying selection. Also, just for the sake of simplicity and time, it's a little easier to interpret. So one last little kind of note before we dive into the results here. I want to step back and kind of mention the context of how these different regions that I'm going to talk about were identified. So again, the enhancers we identified with that cap small RNA sequencing. This is evidence from adipose tissue. So we have some evidence that these enhancers are active in adipose tissue, but we have no idea if they're functional in skin, other tissues in the body, and so on. They might be, they may not be. Promoters, on the other hand, we just simply mine from the genome annotation. Again, if we know where a gene is, we can identify the promoter just upstream of it. So promoters are potentially, or in this analysis, the promoters are potentially regulating genes in all sorts of tissues and cell types. So not necessarily something specific to like lipid metabolism and stuff we're expecting in adipose tissue here. All right, so let's dive into the enhancer results first. This tree is showing the number of enhancer regions, shown with the actual number, the branch length, and the color, just for all sorts of redundancy here. Again, branch specific, so relatively accelerated in a particular branch compared to the rest of the tree. And I've got our hibernating species shown here in bold and with Hib next to them. And just focusing on kind of the immediate patterns, you see a strong signal for acceleration on this ancestral branch, as well as acceleration in the polar bear branch, as well as the ancestor of sloth and sun bear. And looking at the targets of these enhancers that are particularly, evolving rapidly in these branches, or evolved rapidly in these branches, and looking at the KEGG pathways and GO terms that they're involved in, just kind of summarizing these, we see evidence for genes involved in some of these metabolic pathways that we already have keyed into from previous gene expression studies as being important for hibernation, like AMPK signaling, adipocytokine signaling, so adipose-related hormones, insulin signaling, response to nutrient level, GO terms, and more. Similarly, in the polar bear, we see some of these same terms like hippo signaling, as well as terms related to lipid metabolism, adipose homeostasis, and kind of cell-to-cell interaction and communication here. Sloth bear, sun bear, not sure what's going on. We don't see any enriched terms there. It could just be that there's a lot of kind of diverse things happening rather than a concerted signal for particular pathways or terms. But kind of taking this at a kind of broad level, this suggests acceleration of enhancers involved in metabolic gene regulation early on in bear evolution, potentially consistent with kind of the evolution of the bear version of hibernation or specialization of some form of ancestral form of hibernation, as well as rapid acceleration in the polar bear branch, where we know there's been a ton of physiological kind of retooling in a very short period of time here. So contrasting this to promoters, same basic plot here, but for promoter regions, we see kind of a similar pattern. We see an excess of branch-specific acceleration in promoters in the polar bear. Again, this sloth sun bear ancestor, but now also the third strongest signal is for this ancestor of black, brown, and polar bear here. And since these are not derived from the adipose, these are all promoters in the genome, we see some other cool terms that might be relevant for bear biology without doing too much storytelling here, like immune system responses in the ancestor of black, brown, and polar bear. A bunch of metabolism related terms popping up again in the polar bear, including hormone signaling, PI3K, insulin signaling is kind of an interchangeable term for the most part, as well as anti-inflammatory signaling and some cool metabolic process related terms. This branch still is kind of weird. We see a few terms here related to potentially neuronal development and some specific types of metabolism, but still not entirely sure what the story is with that branch there. But something that kind of popped out unexpectedly in this analysis that I think is pretty exciting, which I'm going to actually show you, instead of just summarizing the KEGG and GO enrichment, I'll show you like an actual plot from that. This is for all branches across this tree, and you can probably notice a very apparent trend right here, where eight branches within this tree were significantly enriched for promoters involved in regulating genes in olfactory transduction related processes. So essentially sense of smell. Bears rely on sense of smell for almost everything. They have a very keen sense of smell. They use it for communication between individuals, for foraging, for hunting, potentially for like finding den sites and things like that. So there's been quite a body of ecological and behavioral studies showing how important olfaction is across bears. And some studies that have shown, for example, copy number variation in olfactory genes across different bear lineages potentially associated with that. This is now potentially suggesting that in addition to that, the regulation of the expression of some of these olfactory genes potentially has been kind of a massive signal of regulatory sequence evolution across this group. So zooming back out and again trying to summarize all this, we have evidence for olfactory related promoters throughout a lot of bear evolution in this tree, acceleration of some immune-related genes and some other kind of random terms, it seems, in the sloth sun bear ancestor. And despite, again, these being all promoters in the genome, we're still seeing a pretty strong signal for enrichment of accelerated evolution of promoters involved in regulating metabolic genes that we know are involved or potentially involved in hibernation. So again, this is still kind of the first foray into this work and kind of a first look. There's a lot more that we're doing, but I think this is already suggesting, pretty strong evidence for the role of regulatory sequence evolution as this group is diversified, and particularly in instances like the polar bear here, where we know that the retooling or potential modification or some might argue loss of hibernation has happened really quickly, despite this being a really complex adaptation involving all these complex gene regulatory interactions and more. So something that we're kind of working on currently and planning to do more in the future is trying to do this analysis in the context of protein coding sequence evolution now that we have all these genomes, to try to understand if there are particular terms or pathways or processes that seem to be preferentially driven through the evolution of regulatory sequences versus protein coding sequence evolution and things like that. In the last, I think I'm doing okay on time here, but I want to wrap up quick. I've got a note to be quick here. This is kind of just like a teaser of some cool stuff that we're starting to do and have coming out relatively soon, using population of North American brown bears to provide a unique perspective into our understanding of hibernation as well. So a lot of the work that I've done and others in the realm of hibernation have done have leveraged these different axes of natural variation to piece together how hibernation works, how it evolved. For example, in variation within individuals, like these seasonal shifts in gene expression between the active and hibernation season, variation between species, comparative analyses of species that do and don't hibernate. But I think something that's really been kind of overlooked and really not even looked at all in some cases is the potential for variation within species. Hibernation is often kind of thought of as this binary trait, you hibernate or you don't hibernate. But within species, it's very likely, and I think very possible and very likely, I think, that there is underappreciated variation in response to things such as variation in climate. So this is just showing brown bears here, the current range in green, historic range. They used to go all the way down through Mexico, down in gray. The label got lost somewhere, but I think this is some metric of like seasonality or duration of winter. But you can see even in their more restricted modern range, there's substantial variation in climate in this case, but there's also variation in seasonality, diet, and prey type availability across their range. Things that may impact hyperphagia, getting fat, the timing of which they're entering hibernation, and more. There have been some ecological and some kind of more anecdotal studies that have pointed towards differences in or variation in hibernation timing and duration. So entrance into hibernation, exit from hibernation, varying between different populations, some of them kind of along this latitudinal gradient. And in some other smaller mammals, like some of the ground squirrels, I believe that you can more easily do physiological studies of multiple locations and going out and sampling. There's been evidence for intraspecific variation there, but it has not been looked at in bears. All of this work that I've talked about so far has really relied on that Bear Center facility to get access to these sorts of samples that we want, and it's not feasible, at least now, to go out and sample these same types of samples from bears across this population or their range, especially during hibernation, where in nature they're going to be in dens that are hard to access. But we have a bunch of population genomic data for this group that I'll show you here in a second, and we're trying to kind of work backwards. So instead of understanding whether there's variation, how variation in hibernation manifests across this species, can we look in the genome, look for evidence of selection or differentiation in genes involved in hibernation and metabolism based on all our previous work, and use that to try to work backwards and provide evidence for underappreciated variation in hibernation here. So this work has been driven by Ellie Armstrong at UC Riverside, a former postdoc in the same lab as me and now a assistant professor at UC Riverside, as well as a huge collaborative effort with a ton of US and Canadian wildlife agencies. This is about ready to be submitted, might even be being submitted today, but this really cool study we've been working on for the last few years. The biggest population genomic study of brown bears in North America to date. We have about 350 individuals ranging all the way from the Arctic Circle down through Yellowstone. Not a great sampling in northern Canada, but we're working on that. And there's a lot of really cool stuff. This could be a whole hour or 2 hour talk in itself. But just teasing, there's a lot of really cool perspectives using this data to understand population structure and gene flow across these populations, tracking population declines in Yellowstone and other populations of conservation concern, and assessing things like inbreeding and genetic load as kind of the consequence of that. But using all this data that's been collected from wildlife managers, using hair traps and fecal sampling and things like that, where you can collect genomic data but not the functional data, we're trying to use this to inform our understanding of hibernation. So just to kind of do a quick little method slide, we use this approach called PC Adapt, which is essentially kind of like doing an FST scan between populations, but for all populations at once. So it identifies kind of axes of population structure and variation across all of your samples without having to define specific pairs that you're comparing. And then it looks for loci or SNPs in your data set that are exceedingly correlated with these axes of population structure. So essentially loci that are extremely kind of locally structured throughout this population, with the idea being that these may be involved in local adaptation in particular populations. And we ran this for all of our samples except for the Kodiak Island bears, which you can see are pretty distinct up here. I'll talk about that a little bit more in a few slides. But using every other sample kind of through the diversity or this range-wide sampling here. You get something very similar to a typical Manhattan plot or FST plot where we're looking for, again, the y-axis here being kind of excessive population structure of these loci. Identified a little over 1,000 outlier SNPs. 580 of these were close enough to be potentially in linkage with 203 specific genes. And without trying to do a whole lot of storytelling from a Manhattan plot, we do find some interesting enrichment of these genes in things like insulin resistance, type 2 diabetes, glycolysis, gluconeogenesis, a bunch of other terms that are less easy to interpret. But the thing that I think is really compelling and really exciting here is that when we take this set of 203 genes that are potentially highly structured across these populations and intersect them with previous studies we've done of gene expression between active and hibernation season, over half of these 200 genes have previously been shown to be differentially expressed in adipose, liver, and or muscle. So only 3 tissues, but again, a significant enrichment of these genes for genes that we know are differentially regulated during hibernation, potentially underlying aspects of hibernation itself. So again, kind of a preliminary, but I think exciting kind of first look at this, suggesting that there may be evidence for selection on genes relevant to metabolism and aspects of hibernation across the range of North American brown bears, potentially related to some of these climatic variables that we know range or vary across this range, potentially suggesting subtle and underappreciated variation within hibernation and potentially local adaptation. And a lot of the impetus of this paper is to inform conservation decisions about these small populations, these isolated populations, and try to provide information about what sort of population swaps or reintroductions should be done. And I think that this is kind of an interesting idea here, that there's probably all this physiological diversity that's really not necessarily easy to see or observe or necessarily even detect. In the case of bears, related to adaptations to seasonality in their particular environment and so on, that a bear might look like a good match between location A and location B, but if they're locally adapted to hibernate in that particular environment, that may or may not be a good translocation pair in that case. So more to come from this as well. We're currently investigating selection in all of these non-coding regions we looked at in the previous sections of this talk, doing additional analysis trying to actually correlate some of these these SNPs with aspects of climate like seasonality, temperature during the winter and so on. And we are circling back to kind of tease apart the weird and cool features of this Kodiak Island population, which is shown in pink here. It's a small island system off of Alaska that has been isolated for a very long time. These bears have essentially no genetic diversity, but seem to be doing really well on this island. They are absolutely huge. There's anecdotal evidence for bears just not hibernating and staying out all year. So there's a lot of weird and cool stuff going on here that we're hoping to dive into moving forward. So I think I'm low on time, so wrapping things up, this has hopefully been kind of a crash course in a bunch of cool hibernation biology and progress we've made towards understanding how it works and how it evolved, identifying enhancers underlying this, looking for the co-option of existing regulatory mechanisms involved in hibernation, evidence for regulatory evolution or sequence evolution being important in rapid physiological adaptation, and preliminary evidence for selection on hibernation-related genes across bears. There's a whole lot of other stuff going on I didn't talk about today, using single-cell analysis to understand like cell-to-cell interactions across different tissues, validating a bunch of these enhancer predictions we made using cell culture from the bears, and so on. But to wrap things up, thank you all again for listening. Thank you for the invitation to speak. It's been a joy being here today and seeing these facilities and chatting with a bunch of awesome people. Thank you to many friends and collaborators. There's some familiar faces here in this photo, hopefully. Folks at the Kelly Lab at UC Santa Cruz, where I postdoced, the Washington State University Bear Center, and funding sources. And just for fun, this is a video of a bear-sized respirometry chamber tracking oxygen consumption in bears as they move at different speeds and inclines and so on. This is a horse treadmill, which I guess exists, that we then built a object around. Real quick, my email's up here. Take that down. I'm, again, starting a new lab, trying to not load on too many new collaborations and fun projects on the side just yet, but I'm excited to chat with people if you're interested in this or have research that you think is relevant or that I could be valuable to as well. I'd love to chat more with people that I didn't get a chance to meet with today. And with that, thank you all, and I'm happy to answer questions if we have any time, or happy to stick around after.
All right, that was awesome. Thank you, Blair. I think we have time for like one or two really quick questions. If anybody has any, I'll throw the block at you. Yeah. And by that I mean I'll gently toss it.
Thank you very much. So hyperphagia is crazy.